Hi there, welcome to lesson 7.7. This lesson is about plant feeding and predatory mycophagous or fungivorous nematodes. So what this means is that some fungal feeding nematodes can actually also feed on plants or become predators under certain circumstances. Under certain pressure in their ecosystem, they're able to switch from being fungal feeding to plant feeding or predatory. This is Ditylenchus destructor, and the main character we'll be focusing on in this lesson. So without further ado, let's jump into lesson 7.7. It appears that many mycophagous or fungal feeding nematodes have the ability to become plant feeders. And we often see this in agriculture as a manifestation of plant disease, so they are labeled plant parasites. So both fungal feeding and plant feeding, and particularly root feeding nematodes, have similar structures which enable them to penetrate the fungal and plant cells. And these are named onchostyles, spes, or stomatostyles, and look something like this. It's basically a spear that can be forced through a fungal or plant cell wall. However, plant parasitic nematodes have fungivorous ancestors, and research like this is showing that basically they have evolved or have adapted to eating plants, consuming plants, and in certain situations becoming a problem in agricultural industries. So for example, the genus Ditylenchus is mostly fungivorous, consuming at least 70 species of fungi. And Ditylenchus destructor is the second greatest potato consumer. However, that's provided that Fusarium dry rot, a fungal, so to speak, disease is also present. There's also some evidence that Fusarium will actually attract Ditylenchus destructor by producing specific chemicals that Ditylenchus will be attracted to. And Ditylenchus does grow well on Fusarium as a food source. So it seems to be a combination of factors that cause Ditylenchus destructor to actually be able to consume potato crops in this particular instance. And it's a joint effort where Fusarium seems to be mandatory as an infection in the first place for Ditylenchus to be attracted to the potato crop. So we are treating Ditylenchus destructor as the main cause of the problem. However, research is showing that it is really the fungal problem there in the first place. At the same time, the fungi are there once again as another symptom of conditions created, possibly nutritionally and particularly via the soil ecosystem being out of balance, that makes potatoes vulnerable to the effects that we see when these two organisms come together. However, what's important for us here is that fungal and plant cell wall degrading enzymes exist in some of these nematodes, and therefore, even though they are originally or normally mycophagous or fungal feeding, they also have the capacity through their enzyme production to actually be able to feed on plant material, particularly plant roots, and therefore affecting the crop. And in particular, this relationship between Ditylenchus and Fusarium dry rot also occurs in storage. So temperature plays a very important role, as well as I said earlier, soil conditions and moisture conditions within the soil. So once again, we're coming back to this, what comes first? Is it the disease that's causing the problem, or is it the conditions within which we work and the conditions we create on farm, and particularly in the soil, as to whether a particular organism will manifest symptoms within our plants, because the plants are literally signaling to the organisms, and of course the organisms are signaling to one another, and together can cause what we then name destruction. In fact, Ditylenchus destructor is basically named as such because it is being blamed for the disease. And I'm saying that we can never blame an organism for a disease. It's always associated with symptoms of disease. So basically, there are complex communication systems going on, and they always relate to the actual conditions that allow for that manifestation, that allow for organisms to enter plants, to what we call attack plants, but really the plants are already quite sick in order for them to signal to the organisms to be destroyed. So this is the way I view this system, the soil ecosystem particularly, but any ecosystem, including the human body, can be viewed in a similar way. And of course, this is a beautiful drawing of Ditylenchus destructor combined with photography to show its particular morphological features. And this is another example of the multifunctionality of these fungal-feeding nematodes in the soil ecosystem. So in this study, which was done back in 1995, and it's a very rare study to find, there's only a couple that I was able to fish out of the literature, that suggests that Ditylenchus destructor is in fact a fungal feeder, and only in certain soil conditions it actually becomes plant parasitic. But to backtrack a little bit, Aphelinchus aveni also possesses plant cell wall degrading enzymes, and that's normally for it to cause plant decay, potentially to actually gain access to the fungi that it normally feeds on. However, there are questions around its potentiality as a plant parasite because of these plant cell wall degrading enzymes. So it begs the question whether any of these, so to speak, plant parasitic nematodes are actually plant parasitic, or whether they possess plant cell wall degrading enzymes to gain access to the actual fungi that they normally feed on, which are always associated as a secondary effect of plant disease. But of course, these fungi are also just associated with the plant disease rather than being the cause of the plant disease. So both the nematodes, the so-called plant parasitic nematodes and plant parasitic fungi, seem to me as an effect that is due to the underlying poor soil ecosystem, a result of soil degradation rather than actual causes of diseases. They are literally just there to degrade the plant that is already dying. It's basically decaying initially, and then the signal is sent out for the fungi to help to decay the plant, and then these fungi then attract the nematodes to come onto the scene. But then what we see is the nematode and the disease state of the plant, and blame the nematode, basically determine that the nematode is a plant parasite. But because of Aphelinchus aveni's love of fungi, it is being questioned whether it can suppress Ditylenchus destructor, which is, as we know now, potential plant parasite. So can Aphelinchus aveni be a potential biocontrol for what we call plant pathogenic fungi and plant parasitic nematodes? And in this particular study, they've grown Ditylenchus destructor on Fusarium solani isolated from ginseng roots. So ginseng production is being affected by the proliferation of Ditylenchus destructor, as well as Fusarium solani. So these authors isolated the Fusarium solani, grew it up, and introduced Ditylenchus to grow on the Fusarium solani, and also on the ginseng roots and other plant material. And they found that it preferred to actually grow on Fusarium solani. So it preferred the fungal, in inverted commas, disease than the ginseng itself as a food source. They also added mulched leaves of different plants, including ginseng, to the soil. And this significantly increased Aphelinchus aveni populations and decreased Ditylenchus destructor population growth. So it slowed down the feeding behavior, obviously, of Ditylenchus destructor, potentially by Aphelinchus aveni competing for the preferred food source with Ditylenchus destructor. So these are just some of the interactions that are going on, and it seems to me like we have called something plant parasitic in case of this Ditylenchus destructor nematode, where in fact it's not even interested in the plant as much as it is in already sick plants that are a host for their fungal food source. And there are definitely lots of examples, but mainly in agriculture, of nematodes that are plant parasitic, and that's all they rely on. And we'll cover this in a few lessons. So actual proper plant parasitic nematodes. And maybe that will also bring to light some other information, as is the case here with the Ditylenchus destructor. I don't know. But the more I learn about these different systems and look at various studies, the more it seems to me that the nematodes are a scapegoat and would normally just prefer to feed on fungi if fungi were available, but they're not available in agricultural soils. They have been destroyed by the use of mechanical and chemical agricultural practices, and therefore these nematodes appear to be consuming plants. But it is also questionable whether they're surviving on plants or whether they're actually surviving on the fungi that are associated with sick plants such as Fusarium solani. So this is just to throw a spanner in the works, considering that we're basically spending millions of dollars in trying to control these nematodes as plant parasitic. And in particular, Ditylenchus destructor is named the potato tuber nematode and is associated with huge destruction of potato monoculture, but perhaps it is only just looking for its preferred food source, which are the fungi associated with the sick potato plants. Once again, we've got a beautiful image here of Ditylenchus destructor by Peter Mullen, and the affected potato that hosts the fungi and the nematodes when the conditions are right for that to occur. Here is a more recent study demonstrating that Ditylenchus destructor is in fact a fungal feeder that in certain circumstances becomes a prolific plant-dwelling nematode. The authors of this more recent study in 2020 have recognized that there is lack of information on the actual food source of Ditylenchus destructor and the interactions that this nematode has with fungi in garlic fields. And they also wanted to know whether Aphelinchus aveni, which is a non-parasitic fungivore, as we already know, can control garlic-associated fungi, and therefore whether it can actually suppress Ditylenchus destructor by controlling the fungi upon which Ditylenchus feeds on. They found that most of the fungal species isolated from the garlic plants attracted both Ditylenchus destructor and Aphelinchus aveni, and both of the nematodes increased in population on several different strains of fungi, with some variation between the nematodes as to which fungal species and isolates it preferred to feed on. Aphelinchus aveni suppressed Ditylenchus destructor while feeding on several different species of fungi as a sole food source. In Fusarium oxysporum infested soil, there was a significant suppression of Ditylenchus destructor by Aphelinchus aveni. And here you can see the difference between the garlic bulbs from Fusarium oxysporum infested soil 4 weeks after the addition of Ditylenchus destructor. So A and B shows the top covering of the garlic and the fungal mycelium actually present on the garlic. And then when the top leaves are removed from those bulbs, you can see the damage here. And that's associated with a Ditylenchus destructor infestation. And then when Aphelinchus aveni was added into the mix, there is no damage at all to the garlic bulbs. There's no infestation seen, and Ditylenchus destructor is almost undetectable. So it is obvious that Aphelinchus aveni is a potential biological agent as a control for the fungi associated with garlic plants, as well as the effect of Ditylenchus destructor in conjunction with its feeding effect on the fungi. So these authors determined that the hosts, the true hosts of Ditylenchus destructor are the fungi, and have noted that when garlic seeds are treated with fungicides, Ditylenchus destructor is controlled, but it remains to be elucidated whether the nematode actually feeds on garlic at all, or whether literally it is just a fungal feeder and associates with plants only because it is actually feeding on the fungi that feed on sick plants. So the moral of the story here is that when biodiversity is increased, when fungal-feeding nematodes are allowed to be in the soil, and when the fungi are actually allowed to be in the soil without being treated with fungicides, and the nematodes not being destroyed by nematicides, and the actual soil ecosystem is looked after, and biodiversity is increased by having fungal-feeding nematodes, Ditylenchus destructor is no longer a plant parasitic problem. So it must be elucidated as to whether this so-called plant destroyer, Ditylenchus destructor, whether it is actually the problem, because it seems to me from this study in particular that it is only an issue of soil ecosystem health. And while this is very rudimentary, as there's only 3 players involved in this example, what would happen in a healthy ecosystem where all the organisms are supported and not mistreated with various toxic weapons, and whether that in itself would resolve any of our plant parasitism associated diseases. There's also evidence that plants signal to specific organisms, like for example, Ditylenchus destructor receives a signal from sweet potato plants, and the plant actually attracts Ditylenchus destructor. So there's all this other world of communication going on that we are not understanding fully, and therefore making assumptions and scapegoating various organisms. And I think it would be lovely to see science in agriculture move towards a biodiversity approach and supporting that biodiversity and focusing on the biodiversity in order to deal with disease, or not deal with disease because the plants will be so healthy that no such thing as disease will actually be ravaging our crops. So I do believe that this is a problem of the destruction of biodiversity in soil ecosystems and all ecosystems, where we're seeing the effects of disease as a symptom of destruction of biodiversity. The genus Phyllincus is also a nematode that is able to consume plants as well as fungi. Sometimes these nematodes are called switchers, where they would normally feed on fungi in the soil, but when fungi are not present, they will switch to consuming plants. So therefore they can be described as mycophytophagus. So that means fungal plant eaters. So Phyllincus is considered now a fungivore, but has for some time been considered as plant epidermal cell and root hair feeding nematode, or root parasitic organism. But the genus has been shown, particularly in this study, to feed on all types of what's considered plant pathogenic fungi, so Rhizoctonia solani, Fusarium oxysporum, Pythium ultimum and others, but also saprophytic fungi, so those that just consume dead or dying plant material, but also mushroom-producing fungi as well. And here we have an example of a species in the Phyllincus genus called Micellus, which actually prefers to consume saprophytic fungi as opposed to plant parasitic fungi. And the authors of this particular study actually concluded that it is a fungal feeding nematode, and this was in 2005. So I think before then, this genus was considered to be a plant parasite. So that's just to show what happens in an ecosystem once more with a different group of fungal feeding nematodes, where they are forced to become plant, so to speak, parasitic in situations where fungal biodiversity is not supported and therefore they have nothing else to eat. So it's a survival mechanism, and the authors also conclude that plant parasitic or plant consuming nematodes have actually evolved from fungal feeders, from fungal feeding nematodes, as shown through phylogenetic studies. So it's an adaptation, and particularly in agricultural systems where the organisms are trying to survive on a very low fungal diversity, being forced to consume plants. And another beautiful image by Peter Mullen of Phyllincus tournei, which is a species in the Phyllincus genus. Some of these nematodes that can be termed switches also can become predators, in this case mycophagous predators. So an example of this is Bursa Phalanchus sinensis, where it has been shown that some adult females develop a predatory stylet, lip and pharyngeal glands, including enzymes that allow it to digest the contents of other nematodes and therefore be predatory. They have been shown to pierce Acroboloides species, which is a bacterial feeding nematode, and ingest its contents. However, this predatory behavior is not induced by the potential prey, but in fact by the reduction in their fungal food source, leading to a starvation signal or signals, which then leads to this dimorphism, which results in predatory females being born with predatory mouth parts. In this particular study, the ratio of the predatory to mycophagous nematodes increased in older cultures in nutrient-reduced media. So this dimorphism has been shown to be dependent on the medium in which the nematodes are grown and the fungal species that are provided as the food source for the nematode. And when the predatory females were again fed on fungi, they became omnivores, so they were able to predate as well as to feed on their fungal food source, but their offspring were mycophagous. So as long as fungi are present, the new generation of the nematodes will always be fungal feeding. And this basically just shows the ecological adaptability of these animals. And to me, it signifies just how important they are in the environment, in the soil ecosystem. For them to be able to adapt to eating various food sources regardless of their original role that they were designed for is a testament to their importance in the ecosystem, and their ability to adapt is just phenomenal, that these organisms can literally completely change their diet, their mouth parts and enzymes that allows them to ingest very different food sources and to also be able to pierce through the various walls of plants, nematodes, and fungi. The image on the right shows a comparison of the mycophagous or fungal feeding mouth parts in the female when fungi are available, and females which become predatory when fungi are no longer present for their consumption. And there's a distinct morphological difference in the mouth parts which allow these females to survive as predators in fungal limited situations. We have come to the end of lesson 7.7, and in our next lesson we are turning the tables. Lesson 7.8 will be about nematode eating on nematophagous fungi. So while we've been grilling the subject of fungi being consumed by nematodes or nematodes consuming fungi, in our next lesson we'll look at how fungi consume nematodes. You'll be hearing from me again then in lesson 7.8. Bye for now.